Welcome back, friends. In the previous video we discussed about all of the facts on the anatomy of the C4 plants and about the mesophyll and mesophyll and the bundles with chloroplast. So in this video I want to discuss about the Hatch-Slack pathway and all about C4 photosynthesis. Kwa hiyo Hatch-Slack pathway ni mechanism ya dark reaction kwenye C4 plants. Kama ambavyo tuliona kwenye C4 plants, the first product of photosynthesis is oxaloacetate, while kwenye C3 the first product of photosynthesis is glycerate phosphate. So Hatch-Slack pathway is a pathway transporting carbon dioxide and hydrogen from mesophyll cell to Barnard's cell. Once in Barnard's cell, the carbon dioxide is released again and normal photosynthesis occur. So I want you to discuss or to just look with me this diagram here, which is seen. What is different in Hatch-Slack pathway from the Calvin cycle is what happens in the mesophyll cell, because in the Barnard's cell the Calvin cycle, the normal Calvin cycle occurs. So what happens here that the carbon dioxide fixing molecule here is the PEP. So PEP is the 3-carbon compound. When it fixes carbon dioxide with which is catalyzed by the PEP carboxylase, under the influence of the PEP carboxylase enzyme, oxaloacetate, which is the 4-carbon compound, is formed. So as you can see in the right hand side here, this is carbon dioxide fixation into C4 acid takes place in cytoplasm. So the first product of photosynthesis here is the 4-carbon compound, and then as you can see, this is reduced by oxaloacetate is reduced by the reduced in ADP from light reaction to form malate. So malate is also an acid, a 4-carbon acid. So as we read before the definition of Hatch-Slack pathway, that is the pathway for transporting carbon dioxide and hydrogen from mesophyll to Barnard's. So this is our mesophyll cell and this is the Barnard's cell. So carbon dioxide has already been fixed here, and then hydrogen is added here. So malate has carbon dioxide and has hydrogen, which can be released in the Barnard's cell. So what happens here now? The malate undergo what we call shunt. So this is the malate shunt into the Barnard's cell, and then in the Barnard's cell the malate break down to release carbon dioxide, and also the oxidized in ADP get reduced in the Barnard's cell. So what is the net effect? The net effect is just the transport of carbon and hydrogen from mesophyll cell to the Barnard's cell. So here, as you can see, when the carbon dioxide and hydrogen they are removed, we form pyruvate, and the pyruvate goes back to the mesophyll cell. There the pyruvate is phosphorylated with the 2 ATP molecules to form PEP. So the reaction seems to be like the same, because it is started with the carbon dioxide fixation. Then the second stage can be as malate shunt. Then another step can be the regeneration of PEP, which is carbon dioxide acceptor. So the stages of the reaction can be seen as similar to those of the Calvin cycle, but there is a little difference. So simply the Hatch-Slack pathway is this pathway. From here, if you are going here, you are coming here, then you are coming here, then you are going back there. This is the Hatch-Slack pathway. What is going on here? Carbon dioxide fixation by the Ruby carboxylase, and then Ruby carboxylase to form 2GP. This is just the normal Calvin cycle. However, the efficiency of the Calvin cycle in C4 plants which undergo Hatch-Slack pathway is greater as compared to the efficiency of the Calvin cycle in C3 plants. So all of these reasons we shall discuss later. Why are they so? So in the C4 plants, carbon dioxide fixation occur twice. Here is the first point where the carbon dioxide fixation occur, and here is the second point where the carbon dioxide fixation occur. So all of these factors will be discussed later on the advantage and disadvantages of these types of photosynthesis. So now let's see on the mechanism of Hatch-like pathway. The first stage is acceptance of carbon dioxide or carbon dioxide fixation into mesophyll cell. Carbon dioxide is fixed into cytoplasm mesophyll cell by PEP, and the reaction is catalyzed by PEP carboxylase. So I didn't write here, but here we need a PEP carboxylase enzyme. Here you may write PEP carboxylase. PEP carboxylase. Okay, I know this has brought some of the confusions, but what I wanted to write here is about the enzyme catalyzing the reaction. So I know this has brought some of the confusion to some people, but what I meant is just like this. We have an arrow here, and then PEP carboxylase catalyzes the enzyme to form oxaloacetate. So PEP with the 3-carbon compound, which is phosphoenolpyruvate, and adding carbon dioxide, you will get PEP carboxylase, we will get oxaloacetate, and oxaloacetate is the 4-carbon compound or is the 4-carbon acid. So that's the first stage. Then oxaloacetate is converted to malate, a 4-carbon acid, and is shunted through the plasmodesmata in the cell wall to the chloroplast of the Barnard's cell, where it is converted to pyruvate, a 3-carbon acid, by having carbon dioxide and hydrogen removed. The hydrogen is used to reduce it. So this is still the first step, that the oxaloacetate is converted into malate. Then malate in the mesophyll cell, it is converted into pyruvate. As you can see, this is malate, and the reaction shown here, the conversion of malate to pyruvate, but the conversion for oxaloacetate to malate is not shown. You may see it in the diagram which we was discussing above. Now we have few things to note here. First, carbon dioxide acceptor is PEP instead of BIP of C3 plants, and the enzyme is PEP carboxylase. Now PEP carboxylase is much more efficient than the enzyme of the C3 plants. So why this so that PEP carboxylase is more efficient than the enzymes in C3 plants? We have two reasons. First, it has much higher affinity for carbon dioxide, and the second reason is that PEP carboxylase cannot be affected by oxygen concentration. So first is affinity to carbon dioxide, and the second is that it cannot be affected by the oxygen. The mesophyll cells, carbon dioxide and hydrogen are added. Here in the mesophyll cell, the carbon dioxide and hydrogen are added, and that in the Barnard's cell they are removed again. So the overall effect of this is to move carbon dioxide and hydrogen from mesophyll cell to Barnard's cell. Now the second stage of the Hatch-like pathway is the fixation of carbon dioxide in the Barnard's cells. As I told you that in the Barnard's cell the normal Calvin cycle occurs. So what is required to take place here is the refixation of the carbon dioxide. So carbon dioxide and reduced NAD are produced as well as pyruvate in the Barnard's chloroplast. The carbon dioxide then refixed by RuBip carboxylase in the C3 pathway, and the reduced NADP used to reduce glycate phosphate to sugar. So since every carbon dioxide molecule to be fixed twice, the energy required for C4 photosynthesis is roughly double that of C3 photosynthesis. So that's also the one among the major challenge or disadvantage of C4 photosynthesis, that it requires double as compared to the energy of the C3 photosynthesis. Now note, there are some things to note. First, at first sight, then the transport of carbon dioxide and hydrogen by the C4 pathway seems pointless. So if you don't consider well, you can say that why are the plants doing this, but they get advantage. However, fixation using PEP carboxylase in mesophyll is so efficient that a high concentration of carbon dioxide accumulates in the boundaries. So if you are using the double fixation mechanism, the first fixation fixes a lot of carbon dioxide. This means that the Ruby carboxylase works at an advantage compared with the same enzyme in C3 plants. So the major advantage of this is that the Ruby carboxylase function more efficient as compared to the same enzyme in C3 plants. So there are two reasons for this. Why the Ruby carboxylase works at an advantage. First, because of the high concentration of the substrate, and second is oxygen is competitively excluded from the enzyme by carbon dioxide. So in the Barnes cells, the cells are not in the presence of oxygen. Oxygen is not present there, so that's why the Ruby carboxylase works more efficient. And then the last stage is the regeneration of carbon dioxide acceptor. So the last stage is similar to that of the Calvin cycle. Pyruvate is returned to the mesophyll cell and is used to regenerate PEP by the addition of phosphate from ATP. This requires the energy from two high energy phosphate bonds. So if you have pyruvate here, and then this is phosphoenopyruvate. So if you have pyruvate here, 3 carbon acid, and phosphoenopyruvate is 3 carbon acid phosphate. So you are using 2 ATP to do this reaction, as it has been shown in that diagram. So what are the main advantages of C4 photosynthesis? Main advantage, it improves efficiency of carbon dioxide fixation. It is additional rather than alternative to C3. So C4 photosynthesis adds the end products of photosynthesis, not just only alternative to C3, but it is addition. The second thing, C4 plants are photosynthetically more efficient. Because the rate of carbon dioxide fixation is normally the limiting factor in photosynthesis. So they are more efficient because of carbon dioxide fixation. And the third point, C4 plants grow in region of high light intensity, and they have modified chloroplasts for making more efficient use of available energy. Lakini kwenye disadvantage, C4 plant wana consume energy mara mbili kwenye C4 pathway. Kwa sababu gani? Ya double carbon dioxide fixation. But energy is not normally the limiting factor. Hiyo ndio sababu inafanya C4 ziwe zina. Pia hapa kuna significance of C4 pathway over C3. At the first, C4 plant increase in dry mass more rapidly than C3 plants, and more efficient crops in certain parts of the world. So, but in case of increasing the dry mass, plants undergo different adaptations, and some of them, they are maximum rate of carbon dioxide fixation is greater, and they are more tolerant to dry conditions. So tolerating to dry conditions allow them to continue producing food even in bad conditions. The second point, the optimum temperature for growth of C4 plants is also higher than it is in C3 plants. So the optimal point for the growth of the C4 plants is higher. The third point, plants have closed packed mesophyll cell which provide a small area for better utilization of available water and reduce the intensity of solar radiation. Thus, C4 plants are better adapted to tropical climates. Fourth, they are able to maintain high photosynthesis rate and a low concentration of carbon dioxide. Kwa hizi zinakuwa zinaendelea na photosynthesis vile vile hata kama concentration ya carbon dioxide imepungua kwa sababu ya great affinity of PEP carboxylase 2 carbon dioxide. Yet they do not show photorespiration. Yes, they cannot tolerate saline water because of presence of C4 organic acids. Yes, but they are adapted to high temperature and intense radiation of tropics. So problems faced by the C3 plants as compared to the C4. The first is to obtain sufficient carbon dioxide. And then the second is ruby carboxylase, which is an enzyme. It has problem because it can bind both hydrogen and carbon dioxide, oxygen and carbon dioxide, and if it binds oxygen, then different negative effects occur which are collectively known as photorespiration. We have difference between C3 and C4 and carbon dioxide fixation, carbon dioxide receptor, carbon dioxide fixing enzyme, first product, leaf anatomy, efficiency of photosynthesis. And we have difference between carbon and Hatch-like pathway. As you have seen that in carbon, the first molecule is ruby, where in Hatch-like the first molecule is PEP. So I'm sure this is different. If you understood the mechanism, you can just study this different. In the next video, we shall be discussing about the classulation, acid metabolism, complants. So stay tuned, subscribe the channel, share the link of the channel, share the link of the Telegram group in the description below, and then you shall be providing updates of what is going on day to day. Thank you everybody. Have nice studies.